We're going to do an introduction on the framework of the virtual brain, so-called TVB, and also going a little bit into the virtual brain ontology, what is mean to like define the domain of brain network model for reproducible and comparable computational modeling. So we're going into detail on how those models work, how we set them up, what the mathematics behind are, what the theory behind is, and we go also a little bit into the applications. So we have like 1.5 hours maybe for lectures and then half an hour discussion and questions and answers. But please feel free to interrupt. So maybe you need to speak up. Somehow the window with your face is gone. No. Okay, so just please speak up if you have a question. All right. So, the outlook, I think we're gonna skip maybe some parts if we're really going into detail with the basics. But the general agenda would be of the day an introduction to brain network modeling, um, what about connectomes, local dynamics, how to couple them in the network, how to define this as in a fair manner, a brain network model, how to use also TVBO as a model definition language for the TVB framework. Then how to set up and like first examples and clinical applications and bifurcation analysis maybe, and also maybe stimulating the brain. But you will hear about those things also later. So let's maybe start with the bare introduction. So in order to get a digital copy or yeah, a clone basically of one's brain, one need to look into the head of an individual and create basically a model of the brain. We do this usually with MRI. We construct basically yeah, from a non-invasive imaging technique. Then we also know the wiring, basically the cables. We can also derive this from magnetic resonance imaging, or in animals also from tracer studies. Um, and this is basically the long range wiring, the cables transmitting the information between the areas of the brain, between the neurons. So it's usually we do this with diffusion weighted imaging. So we're reconstructing the flow of water molecules within the brain, and this is a good proxy of um, reconstructing the fiber bundles, so the big axonal connections between the gray matter areas of the cortex and the subcortical gray matter areas. Yes, and this is a static image, right? So how do we really compute the wiring, the information flow in the brain? So how is the information transmitted? How is the information processed on a temporal scale in the brain? So how do we, yeah, measure changes over time? So we can do this also with imaging techniques, but we all those imaging techniques are just proxies for what's actually happening in the brain. So they're actually basically information flow. How is the signal transmitted with the nerve fibers between two neurons? And we can have this in a more conceptual, holistic manner. So also with like philosophy of mind or like the emergence of consciousness, we have our theories how a complex system can like, have an emergent property of those cognitive processes like cognition. And this is based on a complex network, maybe, and also the dynamical perspective of this complex network. So we can basically have a look at the brain with its wiring and connect the wires uh, or have different nodes in the brain, so different locations of the brain areas, and connect them with the wires. So we know how strongly connected on a structural level the nodes are. And then we construct a graph of this wiring diagram, basically, of this network. And this is our perspective of a whole brain network. And with this, we can put some mathematics on top and really try to approximate real empirical brain activity with a mechanistic approach. So you can see this is not a black box. This is based on imaging and construction. We will go into detail also later. So the first question is, why network models? The first argument would be, in clinics, we have sometimes lesions derived from stroke or tumors, and two patients have similar lesion location but different symptoms, or they have similar symptoms and different lesion locations. So we cannot really predict the clinical outcome by the lesion location itself. So we need to have a look at the network. We need to have a look what region or what functional role does this region take within the broader network, and how the information that is now not propagated anymore through this lesion is then disrupting the whole system. So we can see that if there's a lesion in a hub, the whole system is affected in this patient. And if a lesion is affecting only one single region, that it might have less severe consequences on the pathology. So from a clinical perspective, to predict the outcome, we would need to model the whole network, not just a catalog of regions. So like, there's the whole concept of lesion network mapping, where they really try to find out the functional networks underlying certain symptom clusters, which is really well progressed in this domain of research already. So why dynamics? So first of all, we have network, and then we put dynamics on top. So at rest, we have even all the brain areas are active, right? So with the default mode network, we even discovered that there are specific brain networks that are more active at rest and less active during tasks. So it's not just noise. So we have basic structure, spontaneous activity, which fluctuates also. So we have reoccurring patterns. So this brain at rest is never idle. So we have always spontaneous activity, which fulfills a broad range of tasks in the brain. So we can, as I told you, we can reproduce also these default mode or resting state networks across subjects and species. So there must be a fundamental underlying principle of organization. And of course, change over time can not only happen at rest, but it can also happen during tasks or perturbations. So we have a signal processed, an auditory signal processed, a visual cue processed. So we have always ongoing dynamic states. Changes over time in the environment are also perceived and sensed with our senses. And we are never still. So we always prepare also for the next service with predictive coding, for example. So we try to really also adapt to our environment and predict the environment and minimize the error of surprise, basically, in our basic living. So this model of a brain network has not to only to produce the spontaneous activity, but also stimulus responses. And so we can have then a so-called functional connectivity network where we correlate the activities of the different brain regions with each other. And there we can then cluster the different modules or different networks, which are more correlated with each other than others. So we can see here in this mock example that we have 3 clusters of highly correlated areas. And this can be then seen here in this functional connectivity matrix, where we have region by region and the correlation, the Pearson correlation of the time series. So we have then network dynamics, and then also we need to have a look at the time scale. So different measurements have different resolutions of time scale, right? So imaging techniques like EEG or MEG are usually on the faster time scale, or EEG strips, which are also implanted. Then, for example, functional magnetic resonance imaging, bold blood oxygen level dependent signals, which are usually on the range of seconds, which measure basically the oxygenation, so the activity in the brain via oxygen consumptions. And this is acting on a way slower time scale. So we can have different observables, which are only proxies of the actual neural activity, right? So if we place an EEG cap on the head of a subject, um, this measures electrical currents or magnetic currents, right? Or dipoles and depolarization. If we measure with functional magnetic resonance imaging, we measure the magnetic properties of certain ions and molecules. So this is all the observation. We then derive our insights of the actual raw neural activity. So we can also say that this modality is a so-called forward model. So we try to model the observation based on the raw simulated state. Okay, and then we have the time scales with the uh, between milliseconds and seconds. And so the idea is that we have a structural network of our model, then we add on the dynamical perspective, and then also upon of this, we also create a so-called forward model to really model also the observables we try to measure with our imaging techniques. Okay, so this is the basic concept and rationale. We will also get into detail later, but maybe we can show you also the time graph of the history of the virtual brain and the whole framework. So the first public release was in 2013, and we have now 2026, so 13 years it has advanced. It was originally a MATLAB toolbox and then has been translated into Python and is growing with modules and applications in basic fundamental research and clinical applications. So we try to now have fair principles, data standards, gradient based or machine learning based computing. We also try to use digital infrastructures, Bayesian inference modeling, and also now with the emergence of large language models, we also need to adapt basically our frameworks so we can uh, prosper basically from the power of those large language models and help us code, but also help us to validate the models and to really construct the models in a really sound manner. And yeah, so you can also see this with the publication. We have more fundamental mathematical publications from 13, 15, and then we go into the clinical applications with epilepsy, Alzheimer's, DBS. Then we have also more fundamental publications with learning and cognition or traveling waves, infrastructure papers, standardization papers. So it's yeah, a lot going on. And what we've already shown you in the beginning, now we go also a little bit more into the mathematics. We have the connectome, the network, right? The wiring diagram of the brain areas. We have some notion, some model of the local dynamics. We have some notion of how the nodes are coupled, so how much information is transferred to the node based on certain coupling function. And then we have the observations. So in this case, we have a hemodynamic response from a raw neural signal. And the mathematical terminology would be the matrix A with the nodes I, J. So we have one rows nodes from 0 to I, and the other column is basically the same from 0 to J. Usually, we not only measure the weights, so how many cables, how many fiber tracts are between two areas. We also um, get the length of the fiber tracts, so to really geometrically assess how far the signal travels, to compute also delays. So we have also a system of delayed um, information flow. And usually we derive this from diffusion weighted imaging and tractography. Then we have the local dynamics, which is just a function over time, which usually is set up as neural mass or mean field models. And we have coupling, which is also a function of the input and summed over time, summed over the matrix of the nodes. And this is the network propagation, and it also includes the delay. So which proxy state is implemented, so to say, the far away a node is, the far away in time the signal is also integrated. So we have a path signal integrated, basically. And we have an observation function, which is also a transformation, a forward model of the raw neural time signal H. So there's a question. Yes.
Hello, sorry to interrupt. So the coupling is the coupling of the neurons within that neural mass or neural field model, correct? Or
No,
something else?
no. This the coupling is the coupling between the nodes. So you couple. So in this case, you have a network of, let's say, 80 brain areas, and in each region we put in one neural mass model, one model of local dynamics, which have
Okay.
their own dynamics set up with the set of differential equations which describe the
Hmm.
change over time in this node. And the activity of this node is not singular, so it is integrated into the whole network. So the this node's activity influences all connected nodes. And this is defined by the coupling function. We will also take into a look how this coupling function looks like a little bit later. But yeah, the important thing is that we have one node is a local dynamic with a neural mass model, and that's just then interconnected in the whole brain.
Okay, so the coupling is sort of the functional aspect that will be mounted on top of the connectome. So the connectome, the delays and the weights, that's the basically the hardware. It's the what we're getting from the diffusion
Yes.
MRI, for example. Okay. All
Yeah,
right. Thank
exactly.
you.
So the building blocks is diffusion is general MRI to get the geometrics of the brain, to get the cortical folding, to also know where the white matter and the gray matter is. We get the tractograms, the cables. We parcellate the brain into regions. So everything, as I said before. Then we count how many cables are between each parcel of the brain, so we can abstract into the network, because this is the wiring diagram that is then setting up the hardware, so to say. And so then here we can also directly derive the topology of the network with the hubs and clusters. And the output is then the structure backbone of the simulation, which is the matrix region by region, weights and delays. The second building block is the dynamical system we place into each node. We start really with the basics. So, what is a dynamical system? It's a, a system, a physical system that changes over time. So, we have states that can be described with equations, and the change over time can be described with the differential equations. So, we can have, for example, a simple spring, which is a mass attached to a spring. And the change over time is defined by the displacement from the equilibrium, so where we start, and by the velocity. Okay, and so when we pull down the spring, then we displace them. So let's say this degree, and we have a velocity of 0. And with this two set of differential equations, we can compute the change in velocity by two parameters, the stiffness of the spring and the mass of the oscillating body, which gives us the base frequency. And we have x, which is basically the displacement from the equilibrium, right? So if we let them loose, we have a simple oscillation defined by the stiffness and the mass. And this will go on indefinitely with our model. So the only things that can be changed is where we start and how stiff and heavy our object is. So this is a really simple dynamical system, and with two differential equations, or even one second order differential equation, we can define or describe the change over time of this oscillator. Okay, so we already talked about this, so it depends from where we start. And this is the so-called initial condition. So we have the same system of the springs, but this time we change the initial conditions to either -1 below equilibrium, -2, and -3. And all other parameters is fixed, and the velocity initial we have it fixed is 0. So we have the same frequency, but when we let it loose, we see that the amplitude changes. And this is also quite intuitive, but so not only the set of parameters and the model itself, but also the initial conditions change the actual outcome of the change over time. So we see if we prolong the spring longer, it will have a higher amplitude of the oscillation. We can have a look at this not only at the system itself, we can also have a look at it in a so-called phase space. So here we just plot the two states, so position and velocity, against each other. And then we can see what happens depending on where we let the spring loose. So with which velocity and position we start, how the system will behave. And here we see that we have those concentric circles. Depending on the position or the initial velocity, we get a higher amplitude of the oscillation. We also talked about this already, but more to say, or more to show you this also, we can change the parameters as well. So we have the same initial conditions and only change the mass of the spring. And you can see here with a bigger object, it's a higher mass. And from the equation itself, we can compute the frequency of this. So the stiffness and the mass define the frequency. So we can say if we let loose at the same initial condition, the different masses have different frequencies of the oscillating spring. Also pretty intuitive, but maybe to summarize, we have different sets of knobs basically we can tune, and the parameters are the knobs we can explore of one dynamic system. So we can dial in a certain value and the outcoming time series will change. So now we have a model that oscillates indefinitely. So we know that a spring under certain physical conditions does not exhibit a perpetuum mobile in this way. So we can adjust the equations to be more physically realistic. And the same we can do with our neural mass models, which are also dynamical systems, maybe sometimes oscillating systems, to be more physiologically plausible. But now we are physically more plausible with our damping spring. So we add some coefficients, like a constant load and a damping coefficient. So we have a decaying oscillation over time. So while our toy model is indefinitely oscillating, our more complex dynamical system exhibits more realistic behavior dynamics, which is the damping. So the spring will eventually then damp out to the equilibrium, defined also by the mass and the stiffness. Okay, if we look at the phase space again with the damped spring, we have again the spring toy oscillator, which is indefinitely just circulating, orbiting around the equilibrium. And on the right-hand side, we see the damped spring in phase space. We have one attractor, the equilibrium, where no matter where we start, eventually the system will then uh, flatten out and get stable. Okay, any questions for here? Otherwise, we will now get a little bit more into detail. So we now have an overview of the dynamics of one single neuron, maybe, or one single entity. And in the brain, it's sometimes not easy to simulate all neurons at the same time, but or it is not feasible yet to have all the billions of neurons simulated computationally efficiently. So we need an abstraction. And as we see here with our flock of birds, we can quite nicely predict the movement of an individual bird by the movement of the average flock. And this is basically also the theory behind the mean field approach to model brain dynamics on a coarser scale than this individual neuron. So what we all maybe know also from computational basics of computational neuroscience, we have really detailed models of spiking cells of individual neurons. And we can model this as a spike raster. So each neuron is connected with other neurons. So we have here pyramidal cells, excitatory and inhibitory interneurons. They are wired together. And by interacting in this small set of connected neurons and small network of neurons, we can derive the mean activity of the three populations of neurons. And with our mean field approach, we say it is enough to just simulate the mean activity of one cortical column, for example, one lumped representation of individual neurons, because this is enough to predict the activity of an individual neuron. And with this abstraction, we not model thousands of neurons in one cortical column, we just model three populations of neurons, so three average representatives of their individual members of a group of pyramidal cells, for example, in this three population model, excitatory inhibitors and inhibitory interneurons. And as you asked before, so within these neural mass model of one single brain column, we have also coupling, of course, because the neurons itself are also coupled. So we have also influences of excitatory processes and inhibitory processes and an integrator compute node that integrates those signals. So we don't only look at the average activity and not at the individual spikes anymore. And this is how it looks like. So this is the three population Janssen-Ritt model. So we have a set of three second order or six first order differential equations, which model each cell population, like the pyramidal cell, excitatory and inhibitory interneuron. And we derive as the main output of this cortical column, which is basically a difference of the input of the excitatory and inhibitory interneurons to the pyramidal cell. And we have then kind of some functions that convert the input and the state variables and the different parameters. So here we go more into detail. So we don't have a single oscillator anymore. We with one equation and two parameters. We have now more parameters which also model more biophysical realistic processes like the amplitude of the excitatory postsynaptic potential, inhibitory postsynaptic potential, some time constants, maximum firing rate. And we already have also units in a more biophysical, realistic fashion. And we have coupling between the different populations with this C. And what we get out of this single column model is different regimes of brain-like activity. So we have noisy activities, like waxing and waning activity, some alpha or even theta activity. So you can see that with one model, we can exhibit different EEG-like signals. But we're not looking at the one single cortical column or cortical region anymore. As we also saw shown in the beginning, we also look at the different, the holistic perspective with the different nodes and how they are connected with each other. And we show you this now here in a more reduced fashion. So we can have 300 or 1,000 brain regions, or we can have three. So to show you the concept, we just have a look at one visual stream. So we have the primary visual cortex and the secondary visual areas. And so those are nodes connected with the edges we derive from, for example, tractography or diffusion images. So we get this structural connectivity matrix, how strongly the different nodes are connected with each other. So we see, okay, V1, V2 are higher connected than V1 and MT, for example. And we can have this represented either as nodes and edges or as a matrix, but it contains basically the same information. Okay, so we have three nodes now in our system and we don't couple it. So they just run individually, freely. And we have now a more complex or a nonlinear model, which is called the Kuromoto model, but it's also just an oscillator, like our spring model. So we can have the different nodes modeled together in one simulation. But if we don't couple them, they just freely run. And the only difference is where they start. And so they have their phase shifted. This is the only thing. So they are not influencing each other. Once we are add coupling, so we add a term, the input term to a neural mass, here in this case, or to a single dynamical system, we now not only model the change of the state by the base frequency, but also by the input and the base frequency. And we can define how the input is processed here with a difference coupling. And we then see that our network, once it's coupled, it starts to converge, it starts to synchronize. And this synchronicity and desynchronization, these are essential parts of the functional dynamics of a brain network as well, right? So when we look at functional connectivity matrices, we see areas that are more synchronized versus areas that are less synchronized. So how we set up the coupling essentially defines how the brain networks can synchronize and influence each other. So with this basic model, we just reach full synchronicity over time. So we need also to improve there at the end if we want to have a little bit a more detailed representation of actual brain dynamics. So to sum it up, we have the full mathematical framework. So the state over time and the change of it is defined by the function of the local dynamics, the current state, a set of parameters, the coupling input from the connected nodes from maybe an external input over time, plus the noise we add with different noise processes, because we not only model the deterministic part, we can also add some stochastic fluctuations and kind of an error term of all the ground noise a network has. So that's the basic setup of this framework. And then we get evolution of state over time. So to sum it up, we now have also the tractogram, parcellation, brain network, and connectivity matrix A. And the coupling function C, which is just a function of like presynaptic and postsynaptic activity, like processes, and the connectivity matrix as the hardwire. So we have the states as input of the presynaptic of the current, and then the current node we have a look at, and all the distant nodes, all the proxy nodes. And depending on the delay, it's also time lagged, basically signal that is inputted in this presynaptic integration function and the parameters of the coupling function. Yeah, so that's maybe also answering the question of the person beforehand. So we have the integration of all node networks from 0 to j into one input for one node. And this maybe visualize it a little bit more. So we have the distant nodes j, which are summed then together after being processed with the presynaptic function. And then we can also have a postsynaptic function that scales maybe the input to the certain node I. Okay. So how do we define this now? Maybe to put everything or everybody on the same page, what are the fair guiding principles? We try to have findable, reproducible, accessible, interoperable methods and data. So we try to enable replication, reproducibility of those models, because we not only want to show scientific results once, we want to show robust and generalizable results. So we need to find a way to have maybe the same data and the same analysis. So we need to really reproduce a finding that is published by a colleague, maybe. And maybe we have our own data. So we also want to replicate. We have the same data, we have the same analysis and different data. Also, we might use another model easily. So we take the whole framework, the whole setup of a simulation experiment, and just change some parameters of the model or change the model completely. And with the same data, we hope to find similar phenomenons, right? So this is then also a robust result. And we also want to generalize. So we have a different disease, for example, and a different analysis. And we can also use the framework of brain network model to also model different disease pathologies, for example. So in order to have or implement this, we need our model description. So how we define this abstract framework and mathematical framework in a way that is also then programmatically executable. So yeah, so we need to have them findable, so integrating into the web with semantic web or ontologies. These are the things. So a database that you cannot query, for example, is not the best to store your data into, because you need to also make them accessible. So you also need to interoperate between maybe data standards, and also you need to reuse also data, but also methods. So this is the whole in one. So we have a dynamic systems perspective of brain activity. We have a network perspective of brain activity. In some cases, we even have this multi-scale integration. So we zoom into one node and really simulate spiking neurons, or a network of spiking neurons within one node to get the high resolution results Then we project it back to the whole brain network. So this is called a multi-scale approach. We will also hear about this in some of the next lectures. We have observation models. We have also different sets of analyses we can use to have an investigation of how our brain network operates and how different subjects, individual operate. So we have all this we need to somehow structure. And one suggestion is an ontology, so a semantic web structure of defining a domain, in this case, a simulation study, so the domain of brain network modeling in computational neuroscience. And we try to gather all the building blocks to make it reproducible. So we need to find basically an understanding of what is needed to the minimal information needed to make a simulation study replicable or reproducible. So we define the parcellation, the state dynamics, the coupling, the observation, also maybe some explorations, some algorithms we use, some optimization pipelines, some continuation analyses, bifurcation analyses. We need to store also the environment, so which software did we use? Also, if we have a stimulation, TMS, are events where they're watching videos or getting visual cues. This needs to be also defined, and also the way of how this whole dynamical system is computed. So, which is the integration method? What kind of noise have we used? What are the initial conditions? So, these are all the elements that if you change them, your results will be fundamentally different. And this is at the one hand side the beauty of this mechanistical model. So you have many knobs that you can tune that have a specific function in the whole framework, but we need to also be aware of them and also share them if we want to share our results and methods. So we build upon the framework of the virtual brain ontology, which is a domain-specific ontology which models the knowledge of brain network simulation in a way so to make the simulations we construct with the virtual brain to make it findable, accessible, interoperable, and reproducible, and to create basically machine-readable artifacts that we can rerun our experiments and share our experiments, and also have a more intuitive specification of the simulation experiments to model them. So we have a common vocabulary. We just back the terms we were talking about. So network, dynamics, state variable, parameter. We just define them in a common vocabulary in a dictionary. And we can also then add synonyms to those terms. So we disambiguate basically what we are talking about without the need of hardcore unification. So we can use different symbols, different nomenclatures, but they point to one and the same concept. And this is based on semantic web methodologies introduced, for example, by Tim Berners-Lee, one of the founders of the World Wide Web. And it's all about machine-readable metadata. So we try to capture in a compact and user-friendly way a schema that describes your simulation framework, as we discussed before, what the building blocks are, what is needed to define the experiment. And the beauty of this is that we can have now, with this standardized vocabulary and machine-readable metadata, we have specification that is interoperable, and we can also integrate them in a curated set of models and to create a database of models and simulation experiments. And if we have the minimal viable information in order to run the experiment, we also then can just automatically generate code. And with the emergence of large language models, for example, we don't need to bother about how to code the whole system from front to back. We need to worry about what we want to do and what the specifics of our experiments are. So this is also a benefit of using the ontology we can basically also make it accessible for non-programmers. And we now have also then a way of translating one experiment into different backends. So we can generate executable code for TVB, for other frameworks, more machine learning based frameworks like JAX, or more new coding frameworks like Julia, or if you like to also with MATLAB. And how this looks like, you define only the metadata, so the model, the dynamics, what our parameters, what are the values of our parameters, what is the equation of our state variables, how do we want to integrate, and yeah, now in this experiment, we explore the parameter A between in three values, 1, 0, -1, and this is then enough because to generate code with templates that are provided with TVBO. So we define then the whole dynamic system in a specific syntax for a specific simulator like TVB, and then can directly run it. So having this semantic specification language decouples us a little bit from the code implementation. We can just focus on how to define the building blocks and which building blocks we want to integrate in our experiment, and let templates and also at a later stage also large language models do the coding. And we always need obviously the validation, the mm, to the original schema. So we need to find a way to also really constrain our use cases, that we implement exactly what we want to set up. And this is a good way with this metadata schema, which is basically a fixed ground truth of how this experiment should work. So this is the framework of TVBO. We can maybe go into detail a little bit later. So we have the annotation of the different building blocks. We have a database and backends that can be then used. We have a database of also models and experiments that are using the framework of whole brain network model. Then we can implement also other semantic web tools to connect so our studies, our models to more disease-related knowledge graphs, and yes, maybe find also some hypotheses to test with our models. And from defining dynamics to the definition of the network, and to running and analyzing the simulation output, you have a full semantic framework that wraps around the virtual brain. We also have a way of looking at the different models that are already in the database. You can load it into an experiment builder. You can download then the metadata specification. So if you don't know how the specification works, there's an entry point at tvbo.charité.de. It will also show you QR codes in a second. And there you can download the specification of some models and experiments and import them into your own framework. So this is then the specification we have seen just now. And with 2 lines of code, you can import this specification into TVBO, which is just a semantic wrapper of the framework. You then can plot the graphs and visualize the things as we've seen before. And then you can run the experiment as it is in the YAML file, and also render a report. So you can directly render from the semantic specification the actual equations in LaTeX. And that's the benefit of having one single specified metadata schema. You render both the code and the report, the equations, from the same source of truth. So we have no divergence between the code implementation and the actual equations reported in a publication, for example. As you might know, there's often a back and forth with implementation, rounds of reviews that might change the code implementation or change the publication from the original implementation, and then the study does not get reproducible anymore. And this is one safeguard, let's say, to have one source of truth for both code and equations. We also have a graph-based explorer, which is now also published together with our colleagues from Cluj in Romania, CodeMart, which is implemented in a Jupyter Lab environment. So you can directly run also the simulations in your compute infrastructure. There you can look, for example, for schizophrenia and which studies have modeled these. So we have a knowledge graph of studies and clinical diseases or phenotypes that are linked to the studies in PubMed, for example, with the mesh terms. And these studies are linked to certain models that the studies use. So you can really parse the graph and see which models you would like and add them to your workspace and run the simulation directly inside your Jupyter Lab environment. So these are the QR codes. So we have the platform where we have a database of common models, common studies used that are using the framework. There you can import them into your own model builder to customize them. You will be also able to store those models in a website on the platform. So if you create a user account, so you can save for later and adjust those models. You can then import them with the Python package to really get the wrapper for running them in different backends than in your own code environment with TVBO. So you can directly produce the code there. And we have the Jupyter Lab extension that you can find here. Yes, there's a question.
Yes, hello again. So how is this? Well, I mean, maybe that's not the best way to ask the question, but I'm just going to go with it. How is it different between or from the TVB project menu, like the brain simulators? Or is this two different things, or do they talk to each other? One is the backend for the other one?
Yes, so TVB is the simulator. So it's the backend, the actual that runs the simulation. So TVBO is just the framework that tries to summarize all the building blocks in a machine readable fashion, which is also human readable. So it strips away the coding part. But with TVBO, you just load the experiment and then you can export it into TVB code and run it with TVB. So this is the idea. So it's a wrapper,
Oh,
and a
okay.
semantic layer basically, which defines the model. And then we have different backends, but the most prominent and most widely used backend is, of course, the virtual brain, where we then can directly export the code into TVB. And the idea is to have basically a flexible and dynamic user interface and semantic wrapper where you don't need to code. And then from there, you just directly export the code and then you can play around with spare TVB code and run it out also and adjust it, but you don't need to. And TVBO can then, in the end, also ingest again a simulation or a simulator from TVB to make it reproducible to put it back into the metadata schema.
Oh, okay. Thank you.
Yes. Yeah. So yeah, this is the framework, the building blocks. Then we need a specification, and then we need a backend that runs then this model and computes and executes basically the computation. So in short, a brain network model is a collection of nodes that exhibit local dynamics, and they are interconnected within a structured connectome. And we observe the activity through a so-called forward model to compare it then with our observations of empirical studies. And yes, this is again for the question maybe. So we have regions, the connectome, and the forward model. And to the question, it's not the computation software, it's just the framework which harmonizes how the models are specified. So we can share and rerun them. And TVB is then the simulator, which then really computes the results. Okay, since I talked already a lot, and also the next slides will be covered by my colleagues probably also in the next sessions, I will go through the next slides with the applications a little bit more quickly, but I will also share the link with the slides here in the chat, so you can parse the citations and read if you're interested in your domain. So we have applications in basic research. So one or two studies from our colleagues in our lab. One is more focused on decision-making processes. So you can not only model the brain at rest, but you can also model the brain at tasks, or you can also inject basically other models into different brain areas that are then doing more than just resting state network fluctuations. And in this study, there were a basic decision-making model was placed into the network into two brain regions that are competing basically. So we have competing decision-making processes between A and B. And based on the network input, we shift between A and B. And then at some point, the integration has finalized and one has win over the other. So here, Micha could also show that the network structure also predicts what slow and fast it is reaction times relate to the intelligence scores of an individual. So we can really also go into cognitive tasks and decision-making processes. So that high intelligent people take longer for deciding on a hard problem and vice versa. Then on the second or the right-hand side, we can also really model fundamental principles of brain signals and brain organization. So here, Dominic showed that the connectome topology really drives the cortical traveling waves and frequency gradients we can observe in the empirical imaging as well. So this is a really nicely laid out study where we can really show basic fundamental properties of brain activity in the whole network. And I'm gonna rush here really through because I wanted to have a complete overview of what's also been done in the clinical applications, but I'm pretty sure that also my colleague Leon Stefanowski will go there into more detail. But here's I try to give a more thorough overview of what's there. And yeah, so we have a mechanistic approach with individual brain dynamics. So we are going into the domain of personalized medicine with individual digital brain twins, personalized virtual brains. So we can go with a patient scan, create a mechanistic model of disease pathology or even uh, therapy to derive a mechanism we cannot directly measure from empirical studies, or to really go into the mathematical detail of the dynamics of a disease pathology. What is done here already, so this is also part of the ontology. We can analyze which studies use which model and which studies are about which pathology. So we have nervous system diseases, neurological diseases like Parkinson, Alzheimer, MS, and more disorders of consciousness, epilepsy, stroke, or brain tumors, or more traumatic brain injuries. We have studies about mental disorders like obsessive compulsive disorder or schizophrenia or major depressive disorder. And these studies all use the framework of brain network modeling and use different neural mass models and different setups, obviously. But to go through, so we have the virtual epileptic patient, where also now a big pilot clinical study has been run. Now also a big clinical studies with 500 subjects tries to predict the epileptogenic zone, epileptogenic zone with brain network modeling to guide surgery interventions, to guide real surgery, and proved to be really helpful in finding out where a seizure starts in the brain. And there are many studies that have had a look at this. Same for Alzheimer's disease and the dynamics of molecules and how the different proteins that are abundant in the brain drive the network pathology and try to predict the disease and the stage from the network activity simulated. Also, virtual drug applications has been done, for example, by Leon Stefanowski with virtual memantine, where they showed that the slowing that was observed in the model of EEG frequencies could be reverted by giving a virtual drug basically in the brain network model. So that's all a transfer of uh, protein pathology that has an effect on excitation inhibition on individual models. So there's a lot. So please feel free to also explore these studies. We also have studies about schizophrenia and how also excitability and excitatory inhibition, the scaling between both affects also the different pathologies, and this is also quite interesting domain. And also Parkinson's disease, where we not only model the pathology but also virtual DBS. You will hear about this by our colleague probably as well, Jill Meyer, where we try to really model the intervention of deep brain stimulation, where we place an electrode deep in the brain next to the subthalamic nucleus, which relieves symptoms of motor symptoms of Parkinson's disease, and how to model this with the dynamics of the stimulation of an electrode, how the electric field is then propagated in the tissue of the surrounding of the electrode, and then how the different fiber tracts that are passing by are then innervated and passing basically the signal of the stimulus to the target areas in the motor cortex, and how this is then integrated in the whole brain network, and how we can then predict also motor outcome and the placement, the optimal placement and setting of the electrode. This can be also done with this modeling framework. Same with stroke and also brain tumor. So this is both a lesion-based approach where we uh, disrupt or different areas of the network and see how the dynamics change with non-functioning areas or non-functioning connections in a network. Also, we have shown that, or our colleagues have shown that we can model also pathologies in MS or traumatic brain injuries, disorders of consciousness, depression, TMS, so stimulation in depression, how the response of the stimulus, the stimulation invoked potential looks differently in patient groups, and OCD as well. So it's a wide range of diseases, and we can say that we have brain disorders, which are disorders of networks and dynamics. So we have changes in the dynamics, and we can measure and also treat this in with a network-based approach. And it already has reached the clinic with the virtual epileptic patient. So where next? How do we translate also the findings of deep brain stimulation into clinics of Alzheimer and mental disorders? So we can have one framework, one model that explains, predicts, and also rehearses the treatment in silico. And by this, I will skip the next, but maybe there's some questions until now. Okay. Okay. So the last part I want to talk about, since it's also applied in the DBS clinical use case, it's how we stimulate the brain. Because we have the resting state, right? We have a dynamical system. We have the activity of the nodes that are influencing the activity of the whole network. Then we have also stochastic fluctuations. We have noise. But we also have different tasks like sensory input, stimulation input, deep brain electrical stimulation. And we can then probe basically the response of the network and how we can change basically the response and what model we need to use and how we need to parameterize the model to also make the digital twin respondent to input stimulus. So we can also reproduce then evoked responses like those from TMS in depression, but also visual evoked potentials. And we can test this then now in silico before touching a patient and predict the optimal setting. So we have a super simple system, and based on which parameter we take, we can either have exponential growth, so we get into an unstable regime, or we have no change at all, or we go back to the line. So this is just based on a set of parameters. And so we can say, as we talked about already, also about the equilibrium, once we have settled, we can now stimulate the spring or elongate the spring again, so it will oscillate again. And based on how our system is set up, we can have different responses to the stimulus. So a stimulation is a time-dependent event, and we have a symbolic representation of this event with an equation, for example. And this is we can name this, we can have a type, we can have different parameters of the stimulus. So when is the stimulus appearing? Which shape does the stimulus have, which is then governed by the equation? How big is the stimulus? All those parameters can then be set. And we have basically the location where the stimulus enters the system that needs to be defined. And with this setup, the equation, we have also kind of a fair, a reproducible way of describing our stimulus, but we can also use a more complex stimulus to show you in the end. So the recipes would be defining the event, injected at the location where we want to start the stimulus, and then run this simulation. And we start with a simple oscillator model, and it looks like this. So we have a baseline, a damped oscillation. So we like our damped spring. And the input of this is currently 0. So we have a stable focus and a stable equilibrium. After we start at the initial condition, it will just damp out. This is basically our resting state. Then we define the stimulus. This is how it's done in TVBO, in the Python framework, but you can also do this in the metadata framework or with TVB. It's a expression over time. So this is just a simple single pulse. So when we have the time, that is when the time passes the onset, we have an amplitude so long as the width is defined. So this is one single pulse at the onset time of here in 200 milliseconds. But we can also define a Gaussian, a ramp, any shape of the stimulus. And then we define the simulation with the stimulus. So we just add basically to the equation we define of our model, we add the stimulus as a parameter where to inject the stimulus inside our model. And we define the event as it is like this. And then we see our initial damped oscillator that tries to settle to an equilibrium and then gets a huge pulse stimulus and suddenly starts to oscillate until the stimulus input is switched off again, and then it goes back to the resting state. And I skipped this because of time reasons, but we can really analyze this with bifurcation analysis. So to have a look at the regimes a system can exhibit when we iterate through a parameter set. So we iterate through the input parameter of this model, and we can see at some point it starts to oscillate given the input. And there we can really derive how much input this system needs in order to change regime, in order to start oscillating. And this is exactly what we derive here or achieve with our stimulus input. Yes, there's a question.
Yeah, so I just have a question about the scope of the bifurcation analysis that are possible within this framework. So it would be ideal to be able to basically see how different nodes, single nodes in different brain regions are reacting, or the regimes of the reaction, different types of behavior according to different types of input, and also just an overall activity. So do we, like, is it implemented that do we have access to sort of a multi-scale or even multi-dimensional bifurcation analyses in the model, or that's just trying to wrap my head around like what are the available um,
Yes.
possibilities here?
So maybe we just quickly go through the slides of the bifurcation analysis, since it's just important. So we need to we can analyze a brain system in three different ways, right? So we can have look at the time series, at the integration over time. So we have a state that changes over time. It oscillates, it damps. Then, as we've seen, we can have a look at the phase plane. So we can really have a look of the flow. Imagine this as like streams of water, maybe. So this kind of a flushing, right? It goes down a drain. So it we can have this visualization of how the two states interact and where we start, how the system evolves. And we can have a look basically at the bifurcation diagram. So we have a phase plane for each parameter set. Um, so we basically add another dimension, not only the states with one single setting of one fixed set of parameters, we vary a parameter, and then we can see which regimes this system can exhibit. And can we have a stable fixed point, or even do we have oscillations where we just have an unstable uh, regime where basically there's two solutions for one set of parameters. So we can have a linear system, right? This is a super basic linear system of maybe population growth or population decay based on a parameter. So it's either exponentially growing, or it's flatten, or it decays. And as we said, so as if we have a single linear system, we can also derive the solution pretty easily where the equilibrium is. So we see, okay, we have no growth if the parameter a is 0, because then it's just where we start, it's 0. And based on this, we can also derive then the perturbation. This we already have talked about. And now we have then basically different behaviors that can exhibit based on the different parameters. So we can either have a stable node where everything just converges to one fixed point. We have maybe also a saddle node where it really passes by and changes direction. We have a stable focus where we yeah, converge to with the damped oscillation, or we just have a center where we oscillate around. And this is then just one set of parameters, and we can have a look where we at in the bifurcation diagram. And the important bifurcation here would be then the Hopf bifurcation, where we have then a change between oscillating and stable focus. And this we can really have a look in 2 dimensions also. So we switch the parameter and suddenly see that oscillations are exhibited only for a certain, like up to a certain amount of the parameter a here. We can have an oscillation. And this is with then more complex models. We can have a parameter a here, so we have a stable fixed point, and then as we converge, or if we go closer to the Hopf bifurcation, we then really can exhibit oscillation with different shapes based on how we compute basically our different bifurcation diagram. And this is a more complex model. This is the EEG model that has even 2 limit cycles here. So you have then 2 regimes that can have different frequencies, different shapes of oscillations based on the parameter. So this is a way of analyzing what our system can do on a single node. Yes, and yeah, this is how it's done. So you started at an equilibrium and then just slightly increase the parameter and see where we're heading. And we can, with this, we can really try to find a way, if the response is folding back, then we have this branching point and a bifurcation detected. And with this method, we can derive a single node bifurcation, but we can also then basically plot the whole network's response onto the network and see, based on the in-strength gradient here, so how much input the system becomes, is how far right on the bifurcation diagram this node lands. Maybe this answers maybe your question. So we can really also have the network perspective on the bifurcation diagram. So of course, this is not always one-to-one mapped on exactly on this, but we can really derive how each node should behave based on the single nodes bifurcation analysis, and then just use the input, so the node degree basically, as yeah, where we are on the bifurcation diagram. Okay, great. So maybe the last no, seems quite nice from time-wise. So the last minutes we can now talk about how the stimulus now propagates, right? So we have one node where we give a stimulus, and this has then uh, two adjacent nodes connected with the same weight and different lengths. So the delay between those nodes is differently. So we define this as an event with the parameters and the equations. We say, okay, we stimulate node 0 and with scaling of 1. And then we have a short connection and a long connection. And we can really also then plot this three nodes network, and we see the propagation of the stimulus with fast coupling speed, so fast conduction speed. So we have the three nodes. The farther distant away node is obviously peaking at a later stage. But when we also increase the conduction speed, then we also increase the impact of the distance. So we can see that there's a really slow response of the more distant node. So we have the same pulse, the same tracts, but only if we vary the conduction speed, so how fast the signal travels, we have different response curves and different stimulation evoked responses. And we can even have a look at this basic model of secondary stimulus propagation. So we have node 1 connected with node 2 connected with node 3, and only indirectly connected with node 1. And we see also then the secondary polysynaptic propagation of the stimulus. Obviously, it gets uh, transmitted via this mediator interneuron here, for example. So the amplitude also decreases, but we can see that also this whole network has more secondary propagation of those input stimuli. So these are, of course, mock examples of the whole brain network. We just plug out three nodes, see how it works. But this is always a nice way to analyze our models and to set up a theory of dynamic stimulus propagation in a network when we just plug out the three nodes and see, do we have a proof of concept? Do we see the stimulus propagation? Are we in a regime where the stimulus input is even taken on, and how it looks like? So then we can now plug those three nodes back into the whole network. And yeah, maybe first this, those three nodes back into the network and let a more complex musical stimulus input into the auditory cortex, for example. So with TVBO and a certain backend from my colleague, Marius Pille, who is also talking next session, I think. He developed a tool called TVB Optim, which is it's a backend that is centered around Jack's framework for efficiently and gradient-based optimization. And with this framework, we can also load in complex stimuli that are then interpolated. So we just uploaded a short piece of music here, created a network model that has a stimulus input at the primary auditory cortex. And I fitted here the empirical functional connectivity with a super simple process varying one parameter, one global coupling scaling, to see which parameter fits best to the actual empirical FC. We have fairly good correlation here. And then we inject the stimulus and see how the functional connectivity of the whole network changes. And we see here the difference of the listening condition minus the control condition is all the nodes connected to the primary auditory areas here with left and right marked. But also we see differences in secondary network structures of the whole connectome, what the music is basically changing. And we can then plot this also to an the brain network, the brain surface. So we then get the summed input or the sum changes for each region, and we can see how the connectivity changes, how the activity changes. Basically, the input for each region changes based on the musical stimulus. So this is also something we can have a look at, not only the three nodes, but also the whole network response. And one slide back, uh, also we can show really well-studied responses like visual evoked potentials. We can replicate with this stimulus setup a study from 1995 even, which showed that also the Janssen-Ritt neural mass model is capable of showing these different evoked potentials. And this is our replication with the specification of the whole study in one metadata schema of the individual study by Janssen-Ritt in the 90s. Okay, so perfect. Putting it together, so we have one workflow, we have a connectome, local dynamics, we have coupling, we have different regimes of the network. We can fit our model to empirical data. This part I left out because you will hear about this later. So we have now the parameters we talked about, right? So these are different knobs of a system, and we can turn these knobs to see which setting of the different parameters are most closely resembling actual empirical data. And we have stimulation. So the brain network model is based on a structure with weights and delays. It has local dynamics, a coupling function, and a forward model, and observable. We can use TVBO to capture all this and specify this in a machine readable and fair fashion. And we can analyze the different nodes, identify the dynamical regimes. We can also stimulate the brain and turn the model into a mechanistic hypothesis. And with this, we have many clinical applications that can show the use case and also fundamental basic properties of brain dynamics and cognition. Okay, and I want to say thank you to the team that have also working on TVBO and my great colleagues and from our lab at Charité in Berlin, and also our colleagues from Codemat in Cluj in Romania. And also thank you for your attention and all the great questions. So please feel free to reach out if you have any questions. And here are some references for the recording.